So again, I welcome back to Excelsior Academy, and today's topic is going to be the final transport of the bulk molecules. Okay, so today we are going to study about the exocytosis and endocytosis. So till now, in all the topics we were mentioning these names, but how exactly these processes are being taken or taking place inside the cell, we are going to study. It's a small topic but interesting one. So what is exocytosis? Whatever the molecules present inside of the cell, we are going to release outside into the extracellular condition. Now, very good example is about the insulin. So when insulin is released, so it is synthesized, the gene is expressed, the protein is being made, it gets folded into the endoplasmic reticulum. From endoplasmic reticulum, it goes to your Golgi apparatus where the further modification takes place. Insulin gets folded, it has got disulfide bonds, so those happening in the Golgi, then it gets modified, and then in the form of the small vesicles, the vesicles filled with these insulin molecules are then go and bind to the plasma membrane. Okay, so this is how the exocytosis takes place. So we have also discussed about exocytosis when we were talking about the neuronal junctions. Remember the presynaptic, postsynaptic, when we were talking about the ligand gated channels, the acetylcholine as neurotransmitter are getting released into the synaptic cleft from the presynaptic neuron, and then these ligands goes and binds to the postsynaptic. It is the same process of the exocytosis. Okay, so as we were discussing, so these are your Golgi apparatus where the proteins are actually getting folded. Okay, now once they get folded, they go into the vesicle called secretory vesicles. Now these vesicles have got certain signal residues which actually coats them to go and get fused with the plasma membrane. So every time when a vesicle goes and fuses with the plasma membrane, there is an increase in the surface area of the plasma membrane. Do you agree? Okay. So what is happening if suppose your cell is only this much, but every time if suppose the vesicle goes and fuses with this, what is happening? The size of the plasma membrane is increasing. Do you agree? So it's not only one or two vesicles, many vesicles will be going and fusing to the plasma membrane. So eventually what is happening? The plasma membrane, the size will be increasing. But in reality, this is not happening. Why? Because once it has been released outside, the molecules are released outside, the cells will also try to make, they also take out the plasma membrane empty vesicles. Okay. So they form the empty vesicles which goes back and fuses to the Golgi apparatus. So always a vesicle is there which goes and binds to the plasma membrane, and from there the empty vesicles are formed and then goes back to the Golgi apparatus to get fused. So this is how the actual size, the normal size of the plasma membrane of the cell has been maintained. So it's all regulated. Okay. So is it clear? It's very simple. There's as such nothing to remember. What you have to remember is that there are two types of regulation in the case of exocytosis. One is called the unregulated membrane fusion. It is called the constitutive secretion. What do you mean by constitutive secretion? Always. Always needed to be produced. So it means what? There is no further signal and all, but every time it has to just keep on producing. Okay. Then you have got the regulated secretion. Second one. Regulated secretion means sometimes depending on the extracellular signaling. So if suppose ligand goes and binds to the receptor, it gives a signal that I need this particular kind of protein to be synthesized. So then the nucleus synthesizes, ER Golgi modifies, and then those secretory proteins goes and binds. Okay. So what you need to remember is there are two types of exocytosis, constitutive and the regulated one. So let's see in more detail. So we know that the exocytosis, it is a process of active transport. Now, when I say active transport, it means again, it needs ATP. Okay. So all the fusion which is taking place is happening with the help of ATP hydrolysis. So when there is a plasma membrane and a vesicle comes, it has to fuse with the membrane. This fusion of the vesicle is happening with the help of the ATP hydrolysis. And it is also called bulk transport because till now when we were talking about the membrane transporters, at a time only one molecule was going, okay? But in the case of exocytosis, it's bulk, like so many molecules at a time has been released outside the cell. Molecules like neurotransmitters, proteins are transported, secreting them through an energy dependent process. So these, in the case of exocytosis, membrane bound secretory vesicles are carried to the cell membrane and the contents are secreted into the extracellular environment. So this we know. It is possible because the vesicle fuses with the plasma membrane. As I said before, so once they has been fused, the molecules are released, then again the empty vesicle goes back and fuses with the Golgi. So there's one video, I'll send you the link. Maybe I'll put in the description of this. You can watch. Very beautifully, they have shown how exactly the process of exocytosis takes place. I'll explain that also. So now we know that how the exocytosis takes place. Okay. Now, just imagine. I'll just give you the brief idea of how the exocytosis takes place. I'm not sure whether you will get the everything because this concept is clear once you know the entire cell bio. Okay. So if I say that the Cooper book, which I mentioned, is this much big, if you have read the everything, then only the concept that, the video, the imagination becomes clear. So I'll just give you the brief idea. So inside a cell, you have got the cytoskeleton. Okay. The cytoskeleton means there are certain proteins which hold the structure of your cell. Okay, otherwise it will just collapse and become a one dimensional. So your cell is actually three dimensional. It has got all the dimensions. So these cytoskeleton proteins are present everywhere. So it's like a roadways. Okay. So many cytoskeleton proteins are present. So they form like a roadways, like how we have national highways and the roads are there. Now, between these roads, you have got your nucleus, the ER, your Golgi. Okay. Now, from the Golgi, when the vesicle is there, so do you know what and how exactly these vesicles from Golgi to the plasma membrane it is moving? It is not, I agree the cytoplasmic fluid is there. It's not like that the vesicles swim around and by mistake it goes and fuses. Everything is regulated. Okay, so it's not that these organelles are freely floating here and there in the cell. It is not like that. Even a single protein is regulated. Which protein has to go where, everything is decided. Now, how exactly this is happening? There are certain proteins called motor proteins. I don't want you to get confused. So just listen to this. Motor proteins are there called kinesin and dynein. So these proteins are actually carrying these vesicles. Okay. And they move towards the plasma membrane. So these are the motor proteins. So they act like your car, the vehicles. So they take the cargo. So vesicle is your cargo. They take on the top of these kinesin and dynein and they move towards the plasma membrane. If suppose the fusion is inverse, that is endocytosis is happening, they take the vesicle and move towards the center of the cell. So on which they are moving, so these are the those motor proteins are walking on these cytoskeleton proteins. So it's like a roadways are there. You have got these proteins on the top of it, this cargo is there. Okay, whatever the vesicles. So they exactly move like this. So I'll share the video with you. It's the original video, how exactly the protein moves. So if I say that they are moving like this, they actually walk like this. You can see the two legs are there. So they move like this, and on the top of it, it has got the cargo. So one step means one ATP hydrolysis. Another step means another ATP hydrolysis. So you can imagine how many ATP hydrolysis takes place. So it's a really energy dependent process. Okay. Did you guys got little bit idea how the exocytosis is happening?
Yes, ma'am.
Still idea? Okay. So I'll share the video with you guys. Even if you don't get the entire picture, at least some idea you will get. Okay. So this is how the vesicles from Golgi to plasma membrane and vice versa, it is coming. Okay. So there are two types of exocytosis, as just now I mentioned, the constitutive and the regulated one. So the regulated one is also called the calcium triggered non-constitutive. Okay. So it means what? It needs calcium. So exocytosis required an external signal in a specific sorting signal on the vesicles, the Clathrin coat. Now remember in the previous class I mentioned about the Clathrin coat. It is actually used so that it can constrict the plasma membrane like this and help in the vesicle formation. Okay. So from your Golgi, the vesicle needs to be pinched off. Over there we need Clathrin molecules which goes and binds over here and pinches off from here. Okay. So the vesicle is free. Then these Clathrin proteins leave the vesicle and then the vesicle is transferred. Okay. Now, exocytosis in neuronal chemical synapse, etc., interneuronal signaling, it's all because of the calcium. So when we talked about the acetylcholine receptor in the ligand-gated channel, remember I also said that whenever there is an increase in the intracellular calcium, the acetylcholine receptors, the acetylcholine neurotransmitters vesicles are formed. Okay, so it is this the reason. Now, in the case of non-regulated, that is constitutive, it is not depending on the calcium. Okay, so it is performed by all the cells and serves to release of the components of the extracellular matrix or delivery of newly synthesized membrane proteins. So in this case, either you will be secreting uh, the extracellular protein, for example proteases. So they are always released so that they can protect their own cells. You should also know that on your hand, palm, so many proteases and RNases are always produced. And this is why whenever we do any experiments, we need to wear the gloves also. Because on your hands, these proteases, RNases are present. And when you do your Molva experiments, half of the time you don't get the RNA, DNA itself. Okay. And yes, and then they are also responsible for the plasma membrane protein release also. Now how they are doing is, on the membrane itself they will have their receptors present like this. Okay, now we know that these receptors need to be present on the plasma membrane, so these vesicles carrying these receptors will go towards the plasma membrane and fuses. Okay, and only after this fusion, where there is no receptor present and blank vesicle is there, okay, that pinches off and goes back to the Golgi apparatus. Okay, so this is how your exocytosis takes place. So one figure is given, so in which you can see that both constitutive and the regulated one is there. So these are the regulated one where you can have the calcium 2 plus signaling. Okay, then you have got the constitutive secretion. It means such proteins are always required. So one example is your proteases, the other one is RNases. So proteases for protein degradation, RNase is for RNA degradation. And that's why many a times, even though you are exposed to so many viruses at a time, you are not getting a viral infection because the RNases present on our skin is killing the virus, which are weak. Okay. So now what is happening? You get the signal over here. From ER it goes to the Golgi, and from Golgi to early vesicle, then intermediate, then the matured one. Okay. And finally they are secreted out. So depending on the signal, it can be endocrine, paracrine, or autocrine. Yes. Now coming to the endocytosis. So this is the last topic. So endocytosis, this is the receptor mediated endocytosis, wherein which the protein goes and binds to the receptor. So you can see over here. So the receptors are present on the plasma membrane. Okay. to which your ligands or the molecule goes and binds. So now it gives a signal that the coat protein, over here it is clathrin. Okay, so the clathrin, the coat proteins will go and bind to these vesicle forming plasma membrane. So what will happen? Finally, the coat is there and the vesicle is formed. Now this will pinch off from here and it will be taken or inside, whether they will fuse with the Golgi or lysosome depending on the signal. Okay, so this is how the receptor mediated endocytosis takes place. Now we also know that endocytosis, in this case, see, this is I was talking about. So you have got the receptor, to which your ligand goes and binds. Now once this is binding, the adaptin protein first goes and binds to the receptor and it is responsible for bringing the clathrin. Okay, so this one is your adaptin. The dark green is your clathrin. Okay, now you can see that the moment they start binding, there is a vesicle formation. Now this gets pinches off with the help of dynamin. So what are the two new proteins we studied? First one is the adaptin, which is helping in the binding of the receptor as well as the clathrin. Okay, now the vesicle needs to pinch off from the plasma membrane. Okay, so that is done by the dynamin. Finally, when this vesicle is formed, all these clathrin code adaptin will get removed and your vesicle carrying only the receptor as well as the cargo, the ligands will be taken inside. After some time, what will happen? It goes fuses with the endosome or lysosome and only the molecules will be taken and the receptor will be recycled back to the plasma membrane. Okay, so this is how your endocytosis takes place. Again, this is also energy dependent process because at each step, ATP hydrolysis is needed. Okay. Now you know about the phagocytosis and pinocytosis. So phagocytosis is also called where you actually ingest the solid particles. Okay, so entire thing will be taken inside. For example, mainly we see phagocytosis in the case of macrophages. So whenever there is a bacterial attack or any wound healing there is a wound. So we know that whenever there is a wound, the chances of bacterial infection is high. So all these are defense cells like macrophages and all, they come near to the wound area and start acting. Now how macrophages act? By engulfing the entire bacteria. And in this case, it is the phagocytosis. Okay. Whereas in the case of bacteria, it is for eating. So whatever the food source or the nutrients available in the surrounding, they actually engulf the entire thing. Now, pinocytosis. So it is also called the drinking process of the cell in the bulk. So again, it's like, you know, they take the water in large amount and they engulf it. Okay. So is it clear? So thank you so much. If any doubts are there, we can discuss now.